Bismillahirrahmanirrahim. Assalamu alaikum. Our today's topic is about enzymes. Now let's dive in a topic. Enzymes. Enzymes are proteins that act as biological catalysts by accelerating chemical reactions. The molecules upon which enzymes may act are called substrate, and the enzyme converts the substrates into different molecules known as products. Like all catalysts, enzymes increase the reaction rate by lowering its activation energy. Some enzymes can make their conversion of substrate to product occur many millions of times faster. An extreme example is orotidine 5-phosphate decarboxylase, which allows a reaction that would otherwise take millions of years to occur in milliseconds. What do enzymes do? One of the most important roles of enzyme is to aid in digestion. Digestion is the process of turning the food we eat into energy. Examples: there are enzymes in our saliva, pancreas, intestines, and stomach. They break down fats, proteins, and carbohydrates. Enzymes use these nutrients for growth and cell repair. Enzymes also help with breathing, building muscle, nerve function, ridding our bodies of toxins. Now, definition. Enzymes are proteins that help speed up metabolism or the chemical reactions in our bodies. They build some substances and break others down. All living things have enzymes. Our bodies naturally produce enzymes, but enzymes are also in manufactured products and food. There are thousands of individual enzymes in the body. Each type of enzyme only has one job. For example, the enzyme sucrase breaks down a sugar called sucrose. Lactase breaks down lactose, a kind of sugar found in milk products. Mechanism. It's the key. It is actually substrate, and log is enzyme. It is enzyme, and it is active site of enzyme, and it is substrate. This is enzyme substrate complex. The lock and key model portrays an enzyme as conformationally rigid and able to bond only to substrates that exactly fit the active site. The induced fit model portrays the enzyme structure as more flexible and is complementary to the substrate only after the substrate is bound. Because the substrate and the active site of the enzyme have complementary structures and bonding groups, they fit together as a key fits a lock. The catalytic reaction occurs while the two are bonded together in the enzyme substrate complex. Here the catalytic reaction occurs. So enzymes are specific. Now the mechanism of enzyme activity. It is enzyme and it is substrate. Enzymes are specific. It only acts on a specific substrate. It is enzyme substrate complexes form. The enzymatic activity occur, and then the products are formed. It is substrate binds to act. It is substrate and it is binds to active site of the enzymes. Now it is enzyme substrate complex. After the formation of enzyme substrate complex, substrates converts into products. Important digestive enzymes. Number 1, carbohydrates. Number 2, lipase. Number 3, protease. Carbohydrates. Carbohydrates breaks down carbohydrates into sugars. Lipase. Lipase breaks down fats into fatty acids. And protease breaks down protein into amino acid. What are the parts of an enzyme? Each enzyme has an active site. This area has a unique shape. The substance an enzyme works on is the substrate. The substrate also has a unique shape. The enzyme and the substrate must fit together to work. Substrate, and it is active site of the enzymes. Both have unique structure. This substrate must be fit on the specific enzyme. Now, how do temperature and pH affect enzymes? Enzymes need the right conditions to work. If conditions are not right, enzymes can change shape. Then they no longer fit with substrates, so they don't work correctly. Now, number 1, pH. Enzymes are sensitive to acidity and alkalinity. They don't work properly if an environment is too acidic or basic. For example, an enzyme in the stomach called pepsin breaks down proteins. If your stomach doesn't have enough acid, pepsin can't function optimally. Number 2, temperature. Enzymes work best when your body temperature is normal, that is about 98.6 Fahrenheit or 37 degrees centigrade. As temperature increases, enzyme reactions increase. But if the temperature gets too high, the enzyme stops working. That's why a high fever can disrupt bodily function. It is an example of enzymes and substrate. The enzymes glucosidase converts the sugar maltose into 2-glucose sugars. Active sites residues is in red, maltose substrate in black, and NAD cofactor in yellow. It is maltose and convert it into glucose product. Thank you very much. Allah Hafiz.